Every healthy ejaculation releases nearly 200 to 300 million sperm cells, yet only one sperm has the privilege of initiating a new human life.
Is it simply a matter of speed? Or is there a highly coordinated anatomical and molecular selection process best behind it? Fertilization normally occurs in the ampulla because its wide, fluid-filled chamber and lush mucosal folds make it the perfect meeting place for the egg and sperm. Male and female systems work together for one goal. Sperm must travel through the female reproductive tract. Only a single sperm will penetrate the egg's outer layer, marks successful fertilization.
If you are an MBBS, BDS, nursing, or medical student studying embryology, this lecture will take you deep into the anatomy and scientific mechanisms of human fertilization.
Human life begins with fertilization, one of the most remarkable biological events in human development.
From an embryological perspective, fertilization is not merely the union of a sperm and an egg. Rather, it is a precisely regulated interaction involving male and female reproductive anatomy. During fertilization, a haploid spermatozoon, carrying 23 chromosomes, fuses with a haploid secondary oocyte. Each brings 23 chromosomes to produce a diploid zygote with 46 chromosomes. This single cell represents the first cell of a new individual. It subsequently undergoes cleavage, beginning the journey of life. It begins with a single cluster. Hollow blastocyst prepares for life. Cells organize, precision, sparking the creation of every human organ. Therefore, fertilization is regarded as the first and most critical event in human development.
To understand fertilization, it is essential to begin with the anatomy of the male reproductive system. Welcome to today's renal and pelvic anatomy lesson. We start by identifying the ilium, the largest bone of the pelvis. Positioned posteriorly is the sacrum. Finally, at the base, we find the ischium.
The testes are not only responsible for sperm production, but also function as endocrine organs.
Within the interstitial tissue of the testes, Leydig cells synthesize testosterone under the influence of luteinizing hormone.
Testosterone is essential for maintaining male secondary sexual characteristics and for regulating spermatogenesis. Inside each testis are hundreds of highly coiled seminiferous tubules. Their germinal epithelium serves as the site of sperm production. Within the seminiferous tubules, Sertoli cells play a vital role in supporting developing germ cells. They provide structural and nutritional support, form the blood-testis barrier, remove defective germ cells by phagocytosis, ensuring proper spermatogenesis. These cells secrete important regulatory proteins, such as androgen-binding protein and inhibin.
The blood-testis barrier is particularly significant because it protects developing haploid germ cells from the body's immune system.
Spermatogenesis is a continuous process lasting over 70 days. A specialized barrier prevents immune destruction during meiosis. It begins with diploid spermatogonia, which undergo mitotic division to produce primary spermatocytes.
These primary spermatocytes complete the first meiotic division to form secondary spermatocytes.
These cells undergo the second meiotic division. This process produces 4 haploid spermatids.
During spermiogenesis, these spermatids undergo remarkable morphological transformation without further cell division. The nucleus becomes highly condensed, reducing its volume. This forms the acrosome, capping the head. The centriole develops into the flagellum for This transforms the cell into a streamlined spermatozoon.
To facilitate movement, mitochondria condense. They pack densely within the middle piece. This generates the ATP required for motility.
However, sperm released from the testes are not yet capable of fertilization.
They acquire progressive motility and fertilizing ability only after spending approximately 2 weeks in the epididymis, where essential biochemical and structural modifications occur within the sperm plasma membrane.
For this reason, the epididymis is regarded as the principal site of functional sperm maturation.
During ejaculation, Mature sperm travel through the vas deferens by peristaltic contractions and enter the ejaculatory ducts,
where they mix with secretions from the seminal vesicles.
The seminal vesicles contribute nearly 60 to 70% of the total semen volume. Their secretions contain fructose, prostaglandins, amino acids,
and coagulating proteins. Fructose serves as the primary energy source for spermatozoa. The prostate gland then adds an alkaline fluid rich in zinc, citric acid, and prostate-specific antigen, PSA,
which helps neutralize the acidic environment of the female reproductive tract and improves sperm survival.
Finally, the bulbourethral glands secrete mucus that lubricates the urethra and neutralizes residual urinary acidity. Together, these glandular secretions form semen, which has a normal volume of approximately 2 to 5 milliliters and typically contains 200 to 300 million sperms. To understand fertilization completely, the anatomy of the female reproductive system is equally important.
The major organs include the ovaries, uterine tubes, uterus, cervix, and vagina.
The ovaries function both as reproductive organs and endocrine glands. During each menstrual cycle, one dominant Graafian follicle usually develops under the influence of follicle-stimulating hormone.
The luteinizing hormone surge triggers ovulation, resulting in the release of a mature secondary oocyte.
At ovulation, the Graafian follicle ruptures, releasing a secondary oocyte, arrested in metaphase 2 of meiosis.
The oocyte is surrounded by two protective layers known as the corona radiata and the zona pellucida.
Immediately after ovulation, the fimbriae of the infundibulum sweep over the ovarian surface and capture the released oocyte, directing it into the uterine tube. Uterine tube is not merely a transport pathway. It provides the ideal microenvironment necessary for successful fertilization. Its ciliated epithelium, secretory cells, mucosal folds, and coordinated muscular contractions facilitate the movement of both gametes while maintaining conditions favorable for fertilization. The uterine tube is anatomically divided into 4 regions: the infundibulum, ampulla, isthmus, and intramural part. Among these, the ampulla is the widest and most highly folded segment. Its extensive luminal surface area and nutrient-rich tubal secretions provide the optimal environment for sperm-oocyte interaction.
Consequently, nearly 95% of normal human fertilizations occur within the ampulla.
Following ejaculation, hundreds of millions of sperm enter the vagina, but their journey toward the oocyte is extraordinarily challenging.
The acidic vaginal environment, with a physiological pH of approximately 4, destroys a large proportion of sperm. Most of the time, cervical mucus acts as a selective barrier, but during the fertile window, it becomes thin and receptive, allowing sperm to pass through around ovulation. Even after passing through the cervix, sperm encounter leukocytes within the uterine cavity, which eliminate many defective sperm through phagocytosis.
Furthermore, the uterotubal junction is an extremely narrow anatomical passage, allowing only sperm with normal morphology. They need excellent motility to enter the uterine tube. Ultimately, out of approximately 200 to 300 million sperm, only 200 to 300 sperm successfully reach the ampulla. This remarkable reduction is not accidental, but represents a highly civid process of natural biological selection carried out by the female reproductive tract. The purpose of this selection is to ensure that only the healthiest and genetically competent sperm are allowed to approach the secondary oocyte. Nevertheless, reaching the ampulla does not immediately result in fertilization. Before a sperm can recognize the oocyte, bind to the zona pellucida, and penetrate its protective barriers, it must first undergo an essential biochemical process known as capacitation, which prepares it for successful fertilization.
In Part 2, we will explore the molecular mechanisms of fertilization in detail.
Capacitation and hyperactivation prime the sperm. Chemotaxis guide them. They penetrate the corona radiata, binding to ZP3 zona pellucida. The acrosome reaction releases enzymes to enter.
Notice the detailed visualization of the heart's chambers. The pulmonary artery, highlighted here, transports deoxygenated blood.
Subscribe to Zenvi Health. If you found this lecture helpful, please like the video, share it with your medical colleagues, and let us know in the comments which topic you would like us to cover next. In the next part, we will explore the molecular mechanisms that make human fertilization possible.